Welcome back once again. So the last video lecture we talked about the complex process of cellular respiration and how it's constantly ongoing in your cells so that they can harness energy from nutrients and use that energy to make the ATP fuel that they can rapidly use to power things that they need to do. And so how in the world do those types of things in our cells happen so efficiently and in such a coordinated fashion? And that's where enzymes come in. Enzymes are an important topic in any biology class, and there's kind of an old saying in biology that if you don't know the answer to a question on a biology test, you can probably get maybe at least a little partial credit if you find some way to blame enzymes. Enzymes are very, very important. Let's see. All right, come on. There we go. All right, so we've talked about already in the past about metabolism in your body. Metabolism is the sum of all of the chemical reactions that take place in the body. So we've heard this story before. It's not just about cellular respiration. Cellular respiration is one set of a whole bunch of chemical reactions that are used to make your ATP. But of course, there are many other chemical reactions that take place to allow you to stay alive. And those include synthesis reactions, in which smaller molecules are linked together to build bigger ones. Then we have decomposition reactions, where larger molecules are broken apart into smaller ones. And then you have exchange reactions. That's a third type, where some portion of one molecule is transferred over to another one. That's called an exchange reaction when that takes place. Okay, and synthesis reactions, when those occur, synthesis reactions require energy input. We've talked about that before. They're endergonic. Decomposition reactions release energy. Those are exergonic. We can also call synthesis reactions anabolic. Decomposition reactions, breakdown reactions, are catabolic. All right, and you put all these things together and you have metabolism. So like they're showing you here on the diagram, this includes building proteins, breaking down carbohydrates for energy, doing things like sticking phosphates onto ATP molecules. All of these things are ongoing in the body. All right, so that's great. You've got gazillions of chemical reactions that are taking place. And could those things happen randomly? Do you think our cells depend on, for example, if you want to build a protein, you got an amino acid over here, an amino acid over here, and you want to put those two together? Do you think your cells could just rely on those two amino acids bumping into each other and winding up bonding to each other? Well, it could happen. Yes, if two amino acids were diffusing around and they happen to bump into each other, molecules are in motion. Yes, you might get lucky and they might bond together, but it could take a tremendous amount of time to make that happen. We would never be able to survive if we were waiting around for all of these many innumerable chemical reactions that have to take place to happen randomly. We would not be able to survive and maintain homeostasis. So this is where our enzymes come in. Enzymes are catalysts of chemical reactions. And by catalyst, what we mean is they make them happen more efficiently. They speed them up. So what enzymes are actually, most of them are proteins. And they're basically protein machines that speed up or make chemical reactions more likely to occur. So they speed up the frequency of our chemical reactions. Okay, in your body, the cells in your body produce at least 1,000 different kinds of enzymes. I've looked for the number and it is over 1,000. We probably don't yet know the exact number of different kinds of enzymes that are made by the many different cells of our body. But you have many, many, many different chemical reactions that take place, and most of them are going to have some enzyme that forces them to happen. So enzymes are very specific about the chemical reactions that they catalyze, that they force to happen. So this little purple blob we see here is an example of an enzyme, and enzymes, most of them are proteins, not all of them. There are a few exceptions, but by far most of them are proteins, and they have unique three-dimensional shapes. We've learned about how proteins have unique three-dimensional shapes before. And so what they do is they capture whatever it is that needs to chemically react. So if you look here, that's what they're showing you. This enzyme has little pockets in it. That's called the active site, in which it is going to capture things that need to react. Those things that need to react, those things that the enzyme is going to capture, are called its substrates. So that's a term you guys should be familiar with. The substrates of an enzyme are the things that it will capture, it will bond to, and will force a chemical reaction to take place. The example they're showing you here on the slide is that those are supposed to be two amino acids that we're going to bond together. So when the enzyme captures those amino acids, it has captured its substrates. Now that those two things are forced to come together, they're actually going to bond. And yes, they could have randomly bumped into each other and bonded, but when the enzyme is like a magnet for these things. So it captures them, brings them together, and forces that bond to take place. They're showing water being released right there because that is a type of dehydration synthesis, if you remember that from Unit 1. And so now your product over here, that's another term you should know. The product right in there is this dipeptide. The enzyme has forced those two amino acids to be bonded together. All right, so the products of an enzyme, all enzymes have substrates and they have products, or they have a substrate and they will make products. We'll see an example of that in a minute. The substrates are the things that they capture and they're going to do something to. Product is what the enzyme has made, what was released at the end. And notice over here the enzyme releases the product, and now it's available to come back over here and do the same chemical reaction over again. It can capture additional substrates and do another version of the chemical reaction. The little link you see down in here at the bottom of the slide, that will take you to a website from the National Institutes of Health, which is pretty interesting. They've got a nice little, very readable summary about how critical enzymes are for our bodily functions. All right, let's take a look at an example of another type of enzyme. So that was an enzyme performing an endergonic and anabolic type reaction. We were building a dipeptide from two amino acids. Enzymes also perform decomposition and exchange reactions, the other types of chemical reactions as well. All right, so if we start up here we have an enzyme. This enzyme is called sucrase. We'll talk about that in a minute. And here's our substrate over here. Our substrate is sucrose. Sucrose is a disaccharide. It's a glucose and a fructose sugar bonded together. So this is the substrate of this enzyme we call sucrase. So sucrose will fit into the active site. That's a little pocket that binds the substrate. It has a perfect little three-dimensional shape that fits sucrose. So they come together through magnetic attraction. They love each other. And now what happens once sucrose is attached to this enzyme, the enzyme forces the sucrose to break apart. Okay? So the products in this case are glucose and fructose. You see over here, those are your products. All right, so not all enzymes build things. Enzymes also break things down. And in this case, sucrose was broken apart into glucose and fructose. So those are your products. Okay, enzyme names. I want to point out something quickly to you. Many enzymes are named with an ASE on the end, like sucrase is the enzyme that breaks down sucrose. That's a very important enzyme that's involved in our digestion. Sucrose is table sugar, so we take in a lot of table sugar from our diet, and you need sucrase enzymes to split that apart into those two sugars before you can absorb those into your bloodstream and send those off to your cells so that they can provide energy to your cells. Lipase is an enzyme that breaks down lipids. DNase is an enzyme that breaks down DNA molecules. And not all ASE names mean you're breaking down things. There are many examples of those, and some of those ASE ending enzymes are involved in building larger molecules as well. Okay, so we said that the vast majority of enzymes are proteins, and yes, that is true. So we have kind of a picture here of a generic enzyme. And the blue portion on this picture is the protein portion. It's a type of protein. Many enzymes, though, need some additional parts in order for them to work. They can't bind their substrates and do their jobs without some additional parts. And so some enzymes have what we call cofactors, and some enzymes have what we call coenzymes. Some have both, and some have neither. Now they're showing you down here, don't worry about this term apo enzyme, that just refers to the protein portion of an enzyme. But they are showing you here, all right, this is supposed to represent some enzyme. And if you just have that protein portion, the enzyme's not going to work. It can't bind its substrate. When you add in the coenzyme, that's another molecule. It's a smaller organic molecule that attaches to the enzyme, like they're showing you here. And now the enzyme has its correct shape and can bind the substrate and do its job. So what are these things? These cofactors and coenzymes. Cofactors are usually smaller things, and they are usually mineral ions of some type. So things like calcium ions. Calcium has a double positive charge. Magnesium ions. Or maybe you've heard before that humans need small amounts of things like copper and zinc, iron. Remember Fe for iron. Those are examples of things that cofactors can be. They're usually ions of some type. And if you don't have the cofactor there, the enzyme is not going to work without it. So the reason why you need small amounts of certain minerals like zinc and copper and so forth in your diet is because they are cofactors for one or more of your enzymes. Without them, those enzymes would not be able to do their jobs. Coenzymes over here, those are organic molecules, so they're not just ions. And they're often vitamin derivatives. So we all know we need vitamins in our diets. Vitamins are a type of smaller organic molecule. They don't really fit into the protein, carb, lipid, or nucleic acid category. We cannot make them on our own. Our bodies don't make them, but we've got to have them because they are coenzymes for one or more enzymes that do important chemical reactions that help us stay alive. So that's what we mean by cofactor and coenzymes. All right, so again, almost all of your enzymes are proteins. Proteins have many different jobs in the body, and as we've learned before, here's just a review. Proteins have different levels of structure. So the first level here is called the primary structure, and all that is what is the sequence of amino acids. You have 20 different amino acids in your cells that you can use to build proteins. What order are they linked together in, and how long is that chain? That's called the primary structure. Once you have that chain of amino acids, they start to fold up locally into little spirals, or what they call beta pleated sheets. That's what they're showing you over here, which we're not going to worry about that. And then once that happens, once they've folded up locally, those little areas of local folding will all wrap around each other and give you the nice three-dimensional structure. And that's what's absolutely critical for a protein to be able to function properly. And some actually have to get together with other proteins that have three-dimensional structures and make a complex. And so that's a fourth level of structure, sometimes called the quaternary structure. All right, so enzymes are proteins. Proteins have three-dimensional structures, like you see there. And if a protein cannot maintain that three-dimensional structure, it's not going to work. So if it's an enzyme, it's not going to bind its substrates. It's not going to make the products it's supposed to make. And so guess what? If your enzymes, for some reason, don't have their proper three-dimensional structures, you can't do your metabolism. You can't do cellular respiration. You can't build anything. You can't break things down. And your cells are going to wind up no longer functioning. All right, if an enzyme, for some reason, loses its three-dimensional shape, it is said to be denatured. Denatured enzymes don't work. So over here on the left, if that's sort of a representation for how a protein enzyme is supposed to work, if it becomes denatured, it loses that shape, like you're looking at over here. Now imagine if that enzyme has a three-dimensional shape and it's supposed to bind a particular substrate like sucrose. If it looks like this, it's not going to be able to do that. Sometimes, so in that case, the protein is said to be denatured. And if it's an enzyme, you could say the enzyme is denatured. In some cases, if conditions return to normal, that denatured protein might be able to fold itself back up and start working again. In other cases, once a protein is denatured, it's just not going to be able to fold itself back up properly, and it's going to wind up being useless. All right, if your enzymes become denatured, guess what? You are going to die. You cannot survive without your enzymes. All your chemical reactions cannot happen quickly enough randomly without enzymes to allow your survival. Why do we have fried eggs over here? This is because when you see egg whites turn white, what you're actually seeing is as the pan where you're frying the eggs heats up, the proteins in the egg white denature, they unfold. You guys know egg whites have a lot of protein in them. There's a protein in there called egg white albumin, and it denatures and it takes on this white color. And the proteins clump together once they're denatured and give the egg white that consistency that it has. So imagine all of the proteins in your body doing those types of things. That would not be too cool for your survival. All right, what are some things that could make proteins in your body, like enzymes, denature? What are some things? All right, improper temperature, especially heat. Overheating, just like we see over here with the fried eggs. When your body temperature goes much above 98.6 Fahrenheit, your proteins denature, your enzymes quit working, and you're going to wind up dying. So that is why that's why we have to maintain body temperature homeostasis so our enzymes function properly. And actually, if your body temperature drops too low, your enzymes actually slow down. Chemical reactions tend to slow down at lower temperatures, and so that's not good for your survival either. Inappropriate pH. This pen is not writing. My little stylus is not writing too well. That's supposed to be inappropriate. Inappropriate pH. So what do we say the pH of human body fluids is supposed to be? 7.35 to 7.45. Well, guess why that is? Because, and pH is a measurement of the concentration of hydrogen ions you have in a fluid. Guess why that is? If you don't have the proper concentration of hydrogen ions, those hydrogen ions interact with your proteins and other important molecules. If you don't have the right amount, your proteins will denature, and you won't be able to survive. Um, you've also got to maintain proper body fluid concentrations of various electrolytes in order for your proteins to have their proper three-dimensional structures. Otherwise, they'll denature. So all of those different types of things are critical for your enzymes to have their proper shape. And again, bringing back up our little cellular respiration diagram over here from the last video lecture, we talked about how cellular respiration is you've got those three phases there we talked about: glycolysis, Krebs cycle, electron transport chain. Each one of those involves many different enzymes. There are many different steps. It's not a straightforward process to go from glucose and break it all the way down and harness energy to make ATP. There are a number of different enzymes there. And again, if those enzymes become denatured for some reason, heat, inappropriate pH, improper solute electrolyte concentrations, your cellular respiration process is shut down. You're not going to be able to survive. You got to have functioning enzymes. And again, kind of tying things together. So we've got to have, if we're going to do cellular respiration and we're going to have nice, happy enzymes, those things take place in here, inside your cells. Your cells are the places where all the other stuff, like out in here in the extracellular matrix of your tissues, gets made. So your cells are the workhorses. And so we've talked about how then the cells are going to need these different solutes to run those processes. You got to have proper hydrogen ion concentrations to make sure your pH is correct. You've got to get glucose and other nutrients through the blood into these tissue fluids and into the cells so that they can do cellular respiration and harness energy. Then they're going to use that energy to manufacture things like all of these fibers you see over here in your tissues. They're going to build all of the stuff that you see out here so that your tissues have their proper structure and consistency. So think about these different things that we're kind of weaving together here in the early parts of the course, because if you kind of work on that now, it'll make the rest of your A&P experience a little bit easier. All right, we've got one more topic that we're going to discuss get out of my pen mode there. One more topic that we're going to discuss here in Unit 2, and that is going to be a little bit more about how DNA serves as a set of genetic recipes that tells your cell how to make protein. So we've just heard a little bit more about how important proteins are for the functions of your cells. Well, what do we mean by genes on DNA serving as blueprints for those? That'll be the topic for our last video lecture for Unit 2.